Good day everyone, my name is Irene Olores. We are the group 1. Our report for today is all about macroscopic, microscopic ultrastructure, and importance, relevance of wood structure. So I will be the first reporter of macroscopic structure of wood. My topic is growth increments. A tree stem consists of 3 areas: pith, xylem, and bark. The central pith is usually barely visible and does not increase in size through the life of the tree. A cylinder of wood, known scientifically as xylem, the D sapwood plus E heartwood, varies in diameter with age and rate of growth. And finally, the bark sheath can subdivided into inner bark, B, which conduct sugars, and outer bark, C, that serve as the protective layer. New wood and inner bark are added each year by the activity of a layer of dividing cells, a cambium sandwich between the inner bark and sapwood. So, as you can see to the picture, the F refers to the central pith, which means bisan unsa kadugayon sa wood is dili mo increase ang iyang size. Then, ang cylinder of wood, known scientifically as xylem, which is the D or sapwood plus E heartwood that locates in white layer portion of the wood, whereas varies in diameter with age and rate of growth. Then the B in the picture or the inner bark, mao na siya ang magkandak og sugar, and ang C or outer bark serve as the protective layer of the wood. And lastly, the A or cambium, siya ang nagadivide sa cells of inner barks and sapwood. In the north temperate zones, cutting any stem surface will show the wood to be composed of a series of concentric bands. These bands are referred to as growth rings, and in temperate trees, commonly one ring is formed each year. Growth rings actually extend vertically along the stem as a series of concentric cylinders. So, meaning, sa isa ka year or isa ka tuig na siya mo form og ring and extend vertically along the stem as a series of concentric cylinder. Each year, a tree grows in height from its tip. Although new wood is added along the length of the stem, no previous growth rings are present at the top of the stem. The number of growth rings increases on the stem according to the number of annual height increments. The appearance of growth rings is due to changes in the structure of wood produced through the growing season. So, meaning ang kadaghanan sa growth rings mo increase siya according to the number of annual heights sa increments. Then, ang appearance of growth rings, mausab siya due to changes in the structure of wood nga na-produce through sa growing season. Although all trees produce concentric layers of wood, not all trees produce visible growth rings. Neither are all growth rings necessarily annual. In some trees, seasonal changes in wood structure may be so slight that growth rings are not evident. Under conditions of severe drought, an annual growth ring may not be produced. On the other hand, under continuously favorable conditions, such as the tropics, several growth rings may be produced in a year. So, to elaborate the topic, sa temperate forest, temperature is mainly responsible for ring formations, whereas sa tropical trees, ring formation is mainly naga-cause siya by either periodic flooding or sa dry seasons. One of the major functions of wood is to conduct water from the roots to the leaves. However, wood does not continue to serve this function indefinitely. At some stage, the wood cells may become black by the air bubbles, other cells or by deposition of other substances. So meaning, major functions daw sa wood is magkandak og water from roots to dahon, but some stage sa wood cells is magblocking siya by air bubbles, which is makakos og problem sa ato ang wood or kahoy.
Good day everyone, I am Kimberlyn Majestrado. I'm here to tackle about what is heartwood and sapwood, direction in wood, and the differences between softwoods and hardwoods. Heartwood and sapwood. Wood that is functional in water transport is referred to as sapwood, and occupies the outer or more recently formed growth rings. Wood that is no longer functional in conducting water is referred to as heartwood, and occupies the central stem core. So as you can see in the picture, sapwood is the outer light-colored portion of a tree trunk, while heartwood is the central core of the trunk. So in most woods, the heartwood can be distinguished from the sapwood by its darker color. So why heartwood have different color with sapwood? So heartwood is usually dark colored due to the tree's natural aging process and because of the position of organic matter known as extractives, which are responsible for its rich color. However, sapwood is almost always light in color due to the formation of new wood made with living cells. Direction in wood. 3 orthogonal planes are recognized, although the stem can of course be cut in any number of intermediate planes. A horizontal or transverse cut through the stem will reveal the growth rings as concentric circles. The other 2 orthogonal planes of wood are longitudinal. A longitudinal cut in a plane through the pith exposes a radial longitudinal surface. In species with distinct growth rings, this surface will appear to have a series of more or less parallel lines. A board cut to expose a radial longitudinal surface is known as a quarter sawn board. A longitudinal cut in plane at a tangent to the surface of the stem exposes a tangential longitudinal surface. Growth rings here will appear as a series of wavy lines, or cones stacked one above another. A board cut to expose a tangential longitudinal surface is known as a plane, or flat sawn board. So in the picture, you can see the directions of wood, which are the longitudinal, radial, and tangential. So the differences between softwoods and hardwoods. Hardwoods are the most diverse group. They contain both the heaviest and lightest timber examples found in nature. So botanically, softwoods include the conifers that belong to the more primitive group of plants called the gymnosperms. Interestingly, this group of plants is almost wholly composed of trees. Hardwoods belong to the botanical group called the Dicotyledonous angiosperms. This is a very large group of plants, including vegetable and fruit plants, herbaceous flowering plants and weeds, as well as trees. The difference between hardwood and softwood is that hardwood is dark in color, while softwood color is light, and hardwood is heavy, while softwood is lighter weight. So in general, hardwood comes from deciduous trees which lose their leaves annually. Hardwoods refers to trees that have broad leaves such as oak, maple, mahogany, as well as many tropical trees. Softwoods are trees with needle-like or scale-like leaves. Example, pines, spruces, and hemlocks are all classified as softwoods. So most of the softwood trees are evergreen, that they keep their leaves or needles throughout the year, unlike a hardwood leaves lose their leaves annually or yearly. So in hardwoods, the water-conducting cells, known as pores or vessels, are commonly very much larger in diameter than the cells, known as tracheids, in softwoods. The pores can frequently be seen with the naked eye as a number of pinholes in the transverse surface of the wood. As a result, hardwoods are commonly referred to as porous woods and softwoods as non-porous woods. So you can see in the picture between the difference between the softwood and hardwood. So hardwoods are referred as porous woods and softwood as non-porous. And that would be all. Thank you.
Good day everyone, I am Daryl Toralba and I am going to present microscopic structure of wood. This is a scanning electron micrograph of an eastern spruce woodblock, a soft wood. Some cells run longitudinally, but some cells run horizontally. The big hole is called resin canal. The majority of cells shown here are called longitudinal tracheids. On different surfaces, the wood structure appears differently. This unique structure differs from other raw materials like metal, plastic, concrete, and rocks. So this photo on the right side is a scanning electron micrograph, an example of a softwood, an eastern spruce wood block. As you can see, there are more longitudinal tracheids than horizontal. There is also a big hole with an abbreviation of R and C, and that is resin canal. In contrast to softwood, the structure of hardwood is more complicated due to more cell types existing in hardwoods. The large holes represent the vessel elements. The small holes are the fibers. The lines between the vessel on the top of the block are bundles of ray cells called multiseriate cell rays. This is a 3D picture of a birch wood block. As you can see in the picture, there is a large hole called vessel or vessel elements. There is also small holes called fibers and multiseriate rays in which a bundle of ray cells of the lines between the vessel elements on top of the block. As you know, a tree produces a ring annually. This ring is composed of two zones: early wood, light-colored area, longer diameter cells, and the late wood, dark-colored area, smaller diameter cells. The early wood is produced at the beginning of a growing season with a relatively thin cell wall. The late wood is formed late in the growing season with a relatively thick cell wall and small cavity. This picture is a cross-section of redwood. It is enlarged 100 times through a light microscope. This picture also shows that the zones of from the early wood to late wood changes distinctively. This is called an abrupt transition. Some species possess this distinct feature, some are gradual. This is another scanning electron micrograph of a wood which shows the abrupt transition from early wood to late wood. This is a Douglas fir. Red pine. Next, the second one is red pine is another species that illustrates the abrupt change of transition zone. In contrast, some species such as eastern spruce have a gradual change from early wood to late wood. Douglas fir and red pine both illustrates the abrupt change of transition zone, while eastern spruce has only gradual change from early wood to late wood, and that is the reason why eastern spruce is an example of semi-ring porous wood. In balsam fir, the transition zone between the early wood and the late wood displays a gradual change similar to white spruce. Eastern white pine is also shows this gradual transition of cell size from early wood to late wood. As you can see, balsam fir and eastern white pine shares the same gradual transition or change from early wood to late wood. In balsam fir, the thick reddish part is the late wood and below it is the early wood. You can also see that in eastern white pine. In hardwoods, wood structure is more complicated than softwood because there are more cell types. The largest diameter holes in the early wood zone are cross-sectional views of vessel elements. In late wood, these vessel elements are small and sometimes grouped together. Because of this distinctive size and arrangement of the vessel elements, the growth ring is very clear and distinctive. This type of hardwood is called ring porous wood. This micrograph shows a cross-sectional view of red oak. So this is a red oak wood block. According to the statement, the ring porous wood has a very clear and distinctive growth ring. It is because of the distinctive size and arrangement of the vessel elements. For the first picture is an Osage orange. The material inside the vessel elements are called tyloses. Tyloses are ingrowths of adjoining parenchyma or food storage cells into the vessel elements. Tyloses block the flow of water through the vessels. However, in some hardwood, size of vessel elements does not change very much throughout a growing season. A good example of this arrangement is sugar maple. The large circles are the vessel elements. Wood possessing this type of even size vessel elements is called diffuse porous wood. So this is an Osage orange. It is an example of ring porous wood. So diffuse porous wood does not change very much throughout a growing season. From early wood to late wood, it does not change very much. American basswood. American basswood is another good example of diffuse porous wood. Another diffuse porous wood is sweet birch. The vessel elements in this micrograph are solitary, individual vessels, or grouped in multiples of two. In other hardwoods, the size of the vessel elements change gradually from the early growing season to late growing season. This type of wood is called semi-ring porous wood. A good example of this type of wood is walnut. Semi-ring porous wood is the vessel elements of this wood is changing gradually. This is also called slowly growing wood because it grows slowly from early growing season to late growing season. Large vessel early wood is at the bottom of the picture and smaller vessel late wood is at the top. As you have seen in previous slides, hardwood structure is more complicated than softwood. In softwoods, longitudinal tracheids are the major cell type. Therefore, softwood lumber has a uniform appearance. Different softwoods will appear somewhat similar. In hardwoods, because of a greater variety of cell types, appearances are quite different.
Hello everyone, I am one of your reporters, Junelyn Gamayon, and I am going to continue the discussion. So, as you have seen in previous slides, hardwood structure is more complicated than softwood. In softwoods, longitudinal tracheids are the major cell type. Therefore, softwood lumber has a uniform appearance. Different softwoods will appear somewhat similar in hardwoods because of a greater variety of cell types. Appearances are quite different. Different hardwoods may have their own unique and distinctive patterns. The drawing below shows various types of cells arrangement. The large circles represent the vessel elements. The vertical lines represent the ray cells. The little dots are longitudinal parenchyma cells. Parenchyma are food storage cells. Vessels and rays conduct fluids. Now look at the variety of arrangements. So what we have here is the parenchyma configurations occurring in hardwoods as seen in transverse view. So above here is what we call the apotracheal. This one here is what we call the diffuse. the diffuse aggregate, and the banded. Now below here is what we call the paratracheal. This one over here is what we call the scanty paratracheal, unilaterally paratracheal, vasicentric, aliform, aliform confluent, and banded confluent. Now the two below here is what we call the boundary. The first one is what we call the initial, and the second one is what we call the terminal. The next 5 pictures are cross-sectional views of hardwoods that illustrate some of the structural detail that can be seen with a hand lens, 10 to 16 times magnifying glass. Here is an example of various cell types of the cross-sectional surface of a hardwood. You can see the structure through a regular hand lens 16 times. The picture here is what we call the elm. This elm is which is characterized by distinct wavy lines of smaller vessels on the late wood. Oak below is a hardwood that has distinctive wide rays among narrow rays. In the example below, there are 2 white rays. Note also the large diameter early wood vessels and the small late wood vessels that are arranged in flame-shaped groups parallel to the rays. White oak 20 times. In this cross-section of ash above, several rays may be seen, but they are much narrower than those in oak. The late wood vessels are often encircled by fine whitish areas which are groups of longitudinal parenchyma cells. 20 times. The picture in the left side is what we call the yellow poplar. Yellow poplar has small diffuse basal elements and fine rays. The 2 growth ring boundaries in this photo are readily distinguished as whitish lines, again due to the group longitudinal parenchyma cells, 20 times of small diameter. And over here in the right side picture is what we call the birch. Birch is a diffuse porous hardwood that also has fine rays, 20 times. Softwoods. Now you know that the structure of wood is quite complicated, especially the structure of hardwoods. The structure of softwood is much simpler. Here is a close look at pinewood. Most of the cells run vertically and resemble long, straight tubes. These are the tracheids. The circles with a hole at the center side of specimen are bordered pit pairs. These pits are channels through which materials can flow from or into neighboring cells. The black-like holes on top of the specimen are cross-sectional views of tracheids. Wood rays are perpendicular to the tracheids. Now look over here. This one here is what we call the resin canal. And over here, this one is what we call the bordered pit pairs. And next, this one here, the squared look hole, is what they called the tracheids. And lastly, this line over here is what we call the rays. In the left side picture is what we call the southern yellow pine. In this view, southern yellow pine earlywood is light in color, while latewood is darker brown. The large holes are resin canals, abrupt transition 20 times. Next, in the right side is what we call the sugar pine. Sugar pine has large resin canals. The growth ring in the sample is primarily early wood, with somewhat narrow bands of late wood. Gradual transition 20 times. Next is Sitka spruce. This one is what we call the Sitka spruce. Sitka spruce has smaller resin canals than the pines. Note that the canals are located primarily in the late wood. White spots 20 times. This one here is what we call the true fir. True fir has no resin canals. Resin canals are found only in pines, spruces, larches, and Douglas fir 20 times. Now in here, over here, this one is what we call the incense cedar. Incense cedar is a wood with distinctive color and odor like incense cedar smells like pencils, since pencils are made from this wood. And lastly, this one over here is the eastern hemlock. Portions of this eastern hemlock photo shows early wood trichids which are large enough to be seen individually. Look for a very fine honeycomb structure. This one. Honeycomb structure. Larger diameter trichids results in a coarser textured wood 20 times.
Good day everyone, so I am Noemi Montenegro and my topic is all about ultrastructure of wood. So this is a picture of a single softwood trached. So as you can see, the pitch is on the side walls and the lumen is inside the hollow cell. So here are two bordered pits which were observed with a scanning electron microscope. The two pits form a pit pair which is cut open so we can see the inside or pit chamber. Portion of three longitudinal trached lumens or dark areas can be seen here. So here is another structural view of a softwood bordered pit pair. Two tracheids are connected by the pit pair. The overarching pit borders are separated by a central pit membrane, which bisects the pit chamber. As you can see in the picture, a face view of the pit membrane reveals a flat circular disc suspended by strands, much like a trampoline. The disc is called the torus and the strands are known as the margo. The margo is elastic and the torus can move laterally. Here is a pit pair in which this has happened. This sailed type of pit is called an aspirated pit. The pathway between the two cells is black. Pit structure is but one of the ultrastructural features of the woody cell wall. The structure of a cell appears even more complicated if you use the high-powered magnification of transmission electron microscope. The micrograph shows a cross-sectional view of pine trached. The cell wall of trached can be divided into various layers as indicated on the picture. The S1, S2, and S3 layers make up the secondary wall. The PR layer is the primary wall. So the cell wall is composed of a number of microfibrils as indicated by the fine lines in this diagram. A microfibril is a bundle of cellulose polymer chains. Orientation of microfibrils is very specific for each layer. As shown here, microfibrils of the secondary two-layer run more or less parallel to the long axis of the cell, whereas microfibrils of the S1 and S3 run more or less horizontally. Orientation of microfibrils in the primary wall is random. Minute structure of the cell wall largely determines properties of individual fibers as well as wood as a whole.
So I am the next reporter. I am Cherelyn Rendon. So my task is all about the wood chemistry. So if we look at still smaller units of structure, we discover the elemental and organic composition of wood. So there are three major elements of wood. Those are carbon, oxygen, and hydrogen. They are combined in complex molecules that are then joined into polymers. These polymers provide the structural integrity of wood. In addition, wood contains small quantities of other organic and organic compounds. So this is the composition of wood by elements. So the elements carbon percent by weight is 49. The oxygen percent by weight 44. The hydrogen percent by weight is 6. And others, the percent by weight is 1. So that is the composition of wood by elements and percent. So next, the polymers of wood can be classified into three major types: the cellulose, hemicellulose, and lignin. The proportion of the three polymers varies between species. So this is the position of wood by types of polymers. So the polymers, cellulose percent by dry wood 40 to 50. The hemicellulose 20 to 35. Lignin 15 to 35. So when we say cellulose, cellulose is the most important single compound in wood. It provides wood strength. So cellulose is a product of photosynthesis. In photosynthesis, glucose and other sugars are manufactured from the water and carbon dioxide. So glucose is first chemically changed to glucose anhydride by removal of one molecule of water from each glucose unit. These glucose anhydride units then polymerize into long-chain cellulose molecules that contain from 5,000 to 10,000 glucose units. So because of the nature of the bond between adjacent glucose anhydride units, the basic repeating unit of the cellulose polymer consists of two glucose anhydride units and is called a cellulobiose unit. So this is the illustration of cellulose polymers. So cellulose polymers are then arranged in crystalline form where adjacent polymers are bonded together laterally by the hydroxyl groups or OH that occurs in each cellulobiose unit. These lateral bonds are not as strong as the end-to-end bonds that join the glucose anhydride units in long-chain molecules, but they are strong enough to provide the strength of wood and also affect other physical properties. So that's all of my tasks. So the hemicellulose and lignin will be discussed the next reporter.
Application of wood structure. Wood identification is possible with a working knowledge of wood anatomy. Basic knowledge of wood structure is essential in determining the best use for each wood type. Extending the structure concept to the molecular level permits discovery and use of many chemical compounds which may be isolated or synthesized from wood. Thus, a study of structure is a foundation upon which wood science and technology is built and is fundamental for a material science approach to wood as a renewable engineering material. There are many other chemical compounds in wood. They usually make up only a small percent of the total composition of wood, but in some cases can be considerable more. In most cases, these compounds are not an essential part of the structure of wood. One class of compound is called extractive and represents a wide range of classes of compounds. One group of extractives that is important commercially is the oleoresins, from which turpentine and various other oils and rosin are derived. Another group of extractives are polyphenols, which include statins, lupins, nines, and lignans. Other organic compounds include gums, tropholines, fats, fatty acids, and waxes. There are also inorganic compounds in wood. They are generally called ash as a group. Calcium, potassium, magnesium, manganese, and silicon are common elements in wood. Silicon is important because it is abrasive and causes ducting of machine tools. Lignin is a class of complex high molecular weight polymers whose exact structure varies. It is an amorphous, not crystalline polymer that acts as a binding agent to hold cells together. Lignin also occurs within cells holds to important rigidity, like cellulose and hemicellulose. Lignin is made from carbon, oxygen, and hydrogen. However, these elements are arranged differently so that they are not classified as carbohydrates. They are instead classified as phenolics, and the polymer is based on the phenolphthalein unit. Hemicelluloses are a group of compounds similar to cellulose, but with a lower molecular weight. The number of repeating end-to-end molecule is only about 150 compared to the 5,000 to 10,000 of cellulose. They are produced from glucose as well as the other sugars such as galactose, mannose, xylose, and arabinose. Product in photosynthesis. Hemicellulose are thus a mixture of various polymerized sugar molecules. In some cases, the polymers are straight chain like cellulose, but polymers with short side chain are also common. Cellulose polymers are then arranged in a cytosine, from where adjacent polymers are bonded together, literally, by the hydroxyl groups that occur in each cellulose unit. These literal bonds are not as strong as the end-to-end bond that join in the glucose hydrate unit into long chain molecules, but they are strong enough to provide the strength of wood and also affect other physical properties. Thank you.